welcome everyone. Welcome to the front row. My name is Jamie Williamson. I'm a professor in the Department of Integrated Structural and Computational Biology here at Scripps Research, and I'll be your host today. And it's my pleasure to introduce Professor Ardem Patapoutian, who's our speaker this week. And, So there's a good buzz going on in the room. I want to welcome everybody in person. Looks like we have about 300 people here in person, and we have over 3,000 people that are watching virtually. So welcome to all of you. There's a lot going on here at Scripps. I could call your attention to the 3 large digits over on the side of the stage. It's our 100th anniversary. We can trace our roots back to Ellen Browning Scripps, founding institutions that eventually became Scripps Research, the Scripps Hospital, and the Oceanography Institute. So it's an exciting time. We've got lots of activities, so keep your eyes peeled for all of that. Now, today, Ardem's going to talk about sensory neuroscience, and I think one of the things that's so engaging about sensory neuroscience is we can get technical, we'll talk about some real hardcore science, but it's something that everybody can relate to because we spend all day sensing things with various aspects. We've got smell, taste, hearing, vision, and all of these things are somewhat easy to at least understand. So, the sense of taste is probably the most primitive thing, or smell. It's molecules in the environment are interacting with receptors, and that specific molecule is telling your brain, there's food here, or go away from that. And that's pretty easy to understand. Hearing is vibration. Vision, there's photons that come up, and there's special receptors that see light and tell you that what's going on in the environment. And so perhaps the most perplexing sense, and the focus of Ardem's talk, is the sense of touch. How do you feel? How do you sense, I got this thing right under me? What is the trigger? What's the signal that gets sent to your brain, and how is that happening at the molecular level? So the way this happens is through these special mechanoreceptors called PIEZOs. And this is, in fact, the subject for which Ardem was recognized for the Nobel Prize in 2021, which was a wonderful day for him and a wonderful day for Scripps. I found it kind of amusing. His lab kind of had a way of keeping him humble. So one of the things that you get when you get a Nobel Prize at Scripps is you get a parking spot. That's the only way you get one. That's it. And the following year, when Barry Sharpless won his second Nobel Prize, his lab went out and papered his spot, said reserved for double Nobel laureate. So I thought it was hilarious. So Ardem's been duly noted for his accomplishments, and what he's doing now is extending this work, perhaps in maybe an unexpected way, into dealing with pain, and I think he'll be dealing with some of that at the end. Okay, so it's my real pleasure to introduce Professor Ardem Patapoutian, and I will come back at the end, so we'll do a Q&A. So for those of you who are here in the audience physically, you put your hand up, and someone with a runner and a lightsaber will find you, and we can have a discussion that way. If you're in the virtual audience, you can type things in. I have an iPad, and I will get your questions. So at the end, Ardem and I will sit here. I'll ask a couple of softballs for him, and then we can engage with the audience. So without further ado, please welcome Ardem.
Hi, everybody. Thanks for coming. So I want to start by showing you a picture of my, this is not just my current group, it's also all the alumni. I think this was a few years back. Every time the Society of Neuroscience meets at San Diego. We have a little reunion. So this includes my current lab members, alumni, and some of their own students. So that would be my scientific grandchildren, I guess. And I want to start with this because most of the work is done by this wonderful young scientists who we train, but also just work with them. And doing the science with them has been one of the great pleasures of my life. So today, we want to talk about the sense of touch. And I'm here to tell you that your sense of touch is really remarkable. You can sense indentations that are 1/500th the diameter of a human hair. That is quite remarkable. But sense of touch is so rich, it's not just about this discrimination at the nanometer scale, but we rely on what we call affective touch for our emotional health. And these are Coco and Puff, our two little cats when they were kittens. And my favorite sense that Jamie had it out there is sense of proprioception. This is related, it's again mechanosensing, as we'll talk about, but instead of telling you what's happening in your skin, tells you what's happening in your muscles. So from how much your muscles are stretched, you know exactly where your limbs are in space. And I can do things like close my eyes and touch my nose. The reason I can do that is because while I'm growing up, I become more coordinated because you become aware of this sense and implement it. So you might not be sensing the stretch of the muscles, but you have a clear vision of what space you occupy and where your limbs are. And this is called proprioception. I think it's one of the most important senses that we have, but most people don't know about it, partly because you can't shut it off. But the photo that I think emphasizes how important the sense of touch is this one. So this little kid is in the Oval Office and very shyly tells President Obama that some people tell him that his hair looks like the president. And Barack, of course, doesn't skip a beat, bends over and says, "Touch it." So, we often talk about seeing is believing, but clearly touching is believing too. And this picture highlights not just the sensing of the texture, but this affective, emotional part of touch that we talked about. So we're interested in the basics. At the cellular, molecular level, how do you do this? How do you sense touch? All of these sensory aspects, pain, touch, and proprioception, are initially detected by a group of sensory neurons. We call them in the peripheral nervous system because they're not in the brain. They're located, as you can see here, just outside the spinal cord. And they gather information from the periphery and send this information to the spinal cord and the brain. One of the interesting characteristics of these neurons is that they're extremely heterogeneous. So some of your neurons here painted different colors only sense temperature or pain. Other only sense low threshold mechanoreceptors involved in touch, and others are proprioceptors. And so the cell bodies, we call them, is in these DRGs, but they send thin processes to the periphery, like the skin and the muscle, get information and send it to the spinal cord. So if these follow these little wires of these neurons into the periphery, for example, as you might expect, they go different places. So proprioceptors innervate muscle spindles, while many different types of cutaneous skin neurites, they're very heterogeneous again. I'm not going to talk too much about it, but it's beautiful heterogeneity. There are some nerve fibers that only sense vibrations. Others sense your hair growing one way or the other. So you don't, again, sense this individually, but all together you get this sense of touch that we know so dearly, but never think about how it comes through. In addition, some of these neurons innervate internal organs, and this is kind of a new area of sensory biology that a lot of people are excited about that we call interoception, which is sensing of internal organs, and we'll talk about this as well. But my lab was interested at the molecular level. How do you sense a gentle breeze or a cactus pricking your finger? And we knew that at these neuronal ends in your fingertips, there are these specialized proteins called ion channels. And ion channels are very simple proteins. They sit at the plasma membrane, this lipid bilayer that envelopes the cell and communicates with the rest of your body. And what they do, these ion channels, is that they're either closed, which means they're doing nothing, or they open. Once they open, ions flow through. And this is the chemical signal that cells understand. For neurons, this flow of ions will actually depolarize the neuron. It's like an electrical switch that starts the signal transduction that will be sent to your brain. So we knew there were ion channels responsible for this, but what were they was not known. And this is what my lab has been studying the last 25 years. Initially, we studied a group of ion channels called trip channels that sense temperature. But the last 14, 15 years, we've focused on pressure sensing, and as Jamie mentioned, identified and characterizing this PIEZO family of ion channels. So most of the talk is going to be about these piezo ion channels, but I want to show just one slide on the trip channels. So TRPA1, one of these ion channels that we identified, is activated by noxious cold, spicy compounds, and give this burning sensation in the mouth. So there are lots of over-the-counter products or ingredients that you put in your mouth that have this TRPA1 burning. One of them is many of these mouthwashes, including Listerine. Wintergreen oil is the active ingredient, methyl salicylate, the compound that gives that burning sensation. And so next time you're using this product and the burning in your mouth convinces you that you're killing bacteria, just realize that you're activating your pain receptor. That's how you feel that way. If you like sushi, wasabi, mustard oil, horseradish, all of these, that wonderful burning in the mouth and the nose are all due to, again, activation of TRPA1. Even raw garlic has a compound that activates TRPA1. And interestingly, when you bake it, there's a chemical reaction that you don't sense the burning anymore. So years of research after this showed that actually in collaboration with chemists here at Scripps, including our president Pete Schultz and Ben Cravat, and we realized that all of these compounds are reactive compounds. And from these experiments, we figured out that TRPA1 is actually a general sensor of pain. And now, actually, blockers of TRPA1, many biotechs and pharmaceutical companies are working on it as perhaps next medicine for various pain indication and cough. Actually, Genentech has a very active clinical trial right now on TRPA1 inhibitors. So I wanted to make 2 points on this. First, people say it takes a long time to make a drug. In this case, very true. We hope to make this faster, but TRPA1 was cloned in our lab in 2003, and 20 years later, here we are. Hopefully next year we'll find out if this will turn into a new medicine. I also want to emphasize that when we cloned TRPA1, this was curiosity-driven research for us. We're just interested in how you sense temperature and other somatosensory stimuli, and yet this is turning out into an important drug discovery, translational research. So I urge everyone to realize that most of what you hear about medicines of today start by basic science curiosity questions in the lab. So the outline for the mechanosensation part, I'll quickly tell you about how we identified piezos and what it looks like, what the protein looks like. And then I'll summarize its role in somatosensation and interoception. And then a few slides on how we're thinking about translating this work. And then at the end, I just have a few non-data fun slides to share with you. So the work started by a postdoc called Bertrand Coste. He was French, he was from Marseille. He came to the lab to find these ion channels. He went back and he started his own lab now. And this is, about 14 years ago, a bit younger. And to look at mechanosensation in a cell, he came out with this very simple way to look at it. So here's a cell. He puts a recording electrode in the cell so he can monitor its electrical activity. And with the glass probe, just pushes on the cell. This is what it looks like. This is the recording pipette. It's simply pushing on the cell. And if this cell has a mechanically activated channel, you should, again, be able to record this current. So at this point, we took a very reductionist approach. Sometimes in science, you have to reduce the question to a simpler question and then go back to the more complex one. So we wanted to find how you sense touch and pain, but we quickly realized that it's going to be too hard to find it in these neurons that I described to you. So Bertrand looked for cell lines, cells that you can easily grow in a dish, that you can then manipulate. And then once we find it, we realize we can go back to the animal setting and human clinic and figure out what they're doing. Without going into details, he found a cell line that is mechanosensitive, and then using bioinformatics and other techniques, limited to about 300 candidates that could be this ion channel. So okay, we said it was simple. So this is, I'm showing you this data. It's not that complicated. I'll try to explain. So this is, on the x-axis, is the currents that he's recording from these cells. At the top is just a control. So this is the average maximal currents he can get. And then each of these little circles with bars on them are one candidate that he is knocking out, taking the protein out of the cell, and see if they still respond to mechanical force. So to get this kind of error bars, Bertrand had to spend about 2 to 3 days on each candidate. So if you kind of do the math, for about 11 months, he came in, worked full-time, and got negative data one day after another. But he didn't give up, and when he knocked down candidate number 72, which was called unknown family 38 member A, so our genome is full of genes that we don't know what they do, and we give them names like this. When he saw this strong downregulation, he decided to call this gene PIEZO1 from the Greek piezi, meaning pressure. So this is great. This means if you take this protein out of the cell, then mechanosensitivity goes away. This means that PIEZO1 is necessary for function. Biologists love to see things that are both necessary and sufficient. And to do sufficiency, what you do is you take this protein and now put it in a cell that's not mechanosensitive, see if by itself it's enough to do this. And if you do this for PIEZO1, you can see compared to control, just transfecting PIEZO1, you get these massive currents. And this is what the data looks like. These show how much you're pushing the cell. The more you push the cell, the more electrical currents that you can record from these cells. And PIEZO1 had a sister called PIEZO2. They're at the amino acid level, which is what constitutes the protein. They're about 40% identical. Okay, so what do they look like? Most ion channels, because I told you they live at the plasma membrane, they have regions that traverse the transmembrane domain. So they can sit at the membrane and be able to communicate outside to inside the cell. Most ion channels have 2 or 6 of these. PIEZO has 38, which is more than any other protein in our genome. On top of that, they come together as a trimer, forming this massive complex at the plasma membrane. In a collaboration with Andrew Ward here at Scripps, we and other labs have solved what we call the structure of PIEZO, which means we know at clear to near atomic level what PIEZO looks like. So each of the 3 trimers, the protomers, are different color. And as you can see, I'm just rotating the protein now, they actually only interact with each other at one of their ends, which is where the pore is. The rest, all these ribbons that you see, are these transmembrane helices that extend into the membrane and presumably are how you sense tension within the membrane. So Rod McKinnon, a colleague and a collaborator, was the first one to suggest that this unusual bending that you see of this protein might be how it is mechanosensitive. That is, when you stretch the membrane, this structure flattens and you get activation. Indeed, both McKinnon lab and Bailong Zhao's lab, who trained in my lab and runs his own lab now in Beijing, showed that if you take this protein out of the cells and reconstitute them and put lots of pressure on them, you can see flattening of the structure, which is quite a remarkable movement of a molecular machine. So all of this are what we call in vitro, which means you're taking the protein out of the cell, putting it either in bilayers or reconstituting it lipids. So you're looking at a very artificial system. We don't know what PIEZOs really look like in its native environment in cells, and this is just the imagining of how it looks like. So this is the plasma membrane in blue. In purple, these PIEZOs kind of dig in and sit there, and any stretch would open them. But we've become very interested in seeing PIEZO in its native form in cells, and can we do that? So fluorescent microscopy has been improving for decades over the last hundreds of years. And now there's these things called super resolution microscopes that are quite amazing. And so to be able to visualize piezo arms on a cell, you have to have a microscope that has its precision be close to or smaller than the protein itself. So this visually shows you how different microscopes are with respect to their sensitivity and where piezo is. So this is confocal microscope. Most scientists use this on a daily basis. As you can see, piezo is so much smaller than what confocal microscopes can resolve, so this is not going to work. But as I said, microscopes have been getting better and better. SIM, STED, STORM. MinFlux is a new super-resolution microscope designed by Stefan Hell. And Scripps was one of the first places that had this microscope. And it's quite remarkable. And we thought, given its resolution, that we might have a chance. This work is done by Eric Mulhall in the lab. And you really have to be Zen to do this experiment. And he's showing his Zen self in this photo. The experiment itself is actually quite simple. He transfects piezo1 in hex cells, and using click chemistry and artificial amino acids, interestingly, two techniques that were developed here at Scripps, he puts a very small Alexa floor dye at the end termini, at the ends of each blade, and then he simply tries to visualize it. So these pictures, when I first saw, just blew my mind. These are raw renderings of labeled piezo. Each color represents a different arm of the piezo. And Eric was already able to distinguish the 3 blades of piezo1 in a fluorescent microscope, which is quite amazing. So if you're not amazed by me just saying this, let me say that the scale bar is 10 nanometer, and the width of a sheet of paper is 100,000 nanometers. So you have to section a paper into 10,000 smaller pieces to get to this resolution. And he already started getting fantastic results. So he then, after getting this, he averages the interblade distance, and this is what he's plotting. And he shows that this in vitro piezo structure that we had is just under 20 nanometers. But when he looks at what piezo looks like in the cell, there's a couple of interesting surprises. One is that they're actually a bit more expanded because the plasma membrane is putting pressure on the protein. But also, the data is more variable. And this we suspected, but we'd never seen. It just means that the ends of piezos are very floppy and they're moving around, which we think is actually plays an important part in its being mechanically gated. On top of it, if he puts pressure on the cells by giving hypotonic solution, for example, he sees that this interblade distance expands, just like it was hypothesized in vitro. This might not seem like much to you, but it's 30% increase in channel diameter and up to 9 nanometer expansion. Just as a reference, this is as wide as piezo can be, according to theoretical calculations. So Eric's not satisfied by this. He, instead of putting 1 fluorophore, he's putting 7 on each piezo because he has a 1 to 2 nanometer resolution, and he'll be able to do almost structural biology looking at this fluorescent microscopy, and we're very excited about this. So I'm going to switch to what piezos do in animals and in humans. So to do this, we use genetics. We use what we call loss of function and gain of function mutations in animal models, such as mice, which means there's no piezo or too much piezo activity. We've also had lots of clinical data that I'll talk about, and together we can find out what these proteins are doing. So we got very excited when we found that piezo2 is expressed in these dorsal root ganglia neurons that I described to you in the beginning, that they're responsible for touch, proprioception, and pain. So Sanjeev Ranade, while he was in the lab, he's now faculty at Burnham next door. He started all these experiments where he knock out piezos and see what happens. And I show this because it's one of the first behavioral experiments we did, also with Matt Petrus, who's here in the audience. So this is not a very sophisticated test, sorry, Matt. And we call it a tape in the back. So you put a tape on the back of a wild-type mouse, and it doesn't like it, and it tries to get it off. And in the knockout, you put a tape in the back, and nothing happens. See what they give Nobel Prizes for these days? But the phenotype of piezo2 is pretty profound. So this is another touch assay where you're pushing with a little wire on their paw, and if you do it, they raise their paw. This is the wild-type animal. You can see piezo2 knockout animals don't respond at all. How about proprioception? So if you don't have this sense of where your limbs are in space, you'll be very uncoordinated. And when we knock out PIEZO2 in a specific cells, we can see that these mice are extremely uncoordinated. You can see the legs there just have absolutely no control over them. Of course, in all of these mice, we don't do just behavior. We want to know that the deficits that we're seeing is at the initial detection of mechanical force. And this is our collaborator, Katie Wilkinson, going to single neuronal fiber and stretching it, sees massive activity increase in wild type, but not in knockout. Something very interesting then happened. My colleague and friend Alex Chesler at NIH, as well as Karsten Bonneman, actually identified individuals who lack PIEZO2. So they have recessive mutations so that both copies of their PIEZO2 is inactive. And remarkably, just like I showed you for the mice, humans without PIEZO2 do not sense touch and do not have proprioception. And this is a test showing lack of proprioception in one of these individuals. I should also say that before we sequenced their genome, the clinicians didn't have a clue that these individuals didn't sense touch and proprioception. Everyone thought they had a motor neuron problem or muscle problem. Nobody thinks about the sensory aspect of this. But once they sequenced it and realized it's a PIEZO2 mutation, it became very clear that they have the same phenotype that the mice without PIEZO2. So in this movie, you're seeing one of these PIEZO2 deficient individuals asked to walk in a straight line while she is blindfolded. She's harnessed for her own protection. This is something that you and I can do very easily. As you can see, she struggles with it. So piezos are required for touch and proprioception, but also one aspect of pain that I'll talk about at the end of the talk, and also mechanical itch. This is the work of Rose Hill in the lab. You know, mechanical itch is when you have a wool sweater that itches or a bug crawling on your skin. Without this, mice don't sense, without PIEZO1, mice don't sense mechanical itch. But over the years, we've also gotten very interested in this idea of interoception. So much less is known about your sensing of internal organs. And this molecule is helping us understand new physiology and biology. So these are collaborations with Alex Chesler at NIH and Steve Lieberlitz at Harvard Med School. We've shown, for example, that without PIEZO2, your control of sensing of your breathing doesn't occur normally. So this is not something you're aware of, but with each breath, you're sensing how much your lung is being stretched and regulating your breathing. And indeed, individuals without PIEZO2 get hospitalized mainly during perinatal times because of breathing problems. You have many ways to control blood pressure. There are specialized neurons that sense in the aorta how much the aorta is stretched. From this, they regulate blood pressure. And we've shown now that in the absence of both PIEZO1 and 2, your blood pressure regulation is compromised. Now, urination is something you're very familiar with, and it's conscious, unlike the respiration and blood pressure sensation. Every time you go to the restroom, you know because your bladder is full. Now that I tell you, it's obvious, right? Because it's very mechanical. As your bladder fills, it stretches, you sense it, that's why you go. For example, again, in the PIEZO2 deficient individuals, they don't have this sense. And so while they're growing up, they keep having accidents, and then their parents, and they come up with a plan where they go to the restroom 4 or 5 times a day to avoid accidents. And now we've taken that and brought it back to the lab and understood where the sensor is, what cells they're expressed in, and what are they doing exactly. Finally, again, not very conscious of this, but your gut constantly moves to help digestion. And this mechanical force is very important. And Rocio Servin in the lab found that without PIEZO2, gut motility is compromised. She did it in animal models, but we found out that humans without PIEZO2 have the exact same issues without it. So all of this is great, but it's just the tip of the iceberg. There's so much to learn in this interoception field. I give you one example. When you eat a large meal, you feel very full, right? That sounds, again, very mechanical. But all of the research in satiety, in energy sensing, is done on chemicals, proteins, fats, and sugars. We don't really know how much the stomach stretch contributes to how much you eat, when you eat, diabetes, obesity, etc. And these molecules, again, are allowing us now to go and ask these questions of how much the stretch is controlled and regulates all these phenomena. And Kara Marshall, who was in the lab and started her own lab at Baylor College of Medicine, is focusing on this question. So till now, I would say everything I told you was, at least scientists knew about how pressure sensing was important for these aspects. But there's many more that we have no clue. Sensory neurons innervate the pancreas, adipose tissue, fat, kidney, and we almost have no idea what they do in these organs. To tackle this, Yu Wang, who was a joint student between my lab and Li Yi lab, as you can see from the photo, she just graduated doing her postdoctoral fellow with Carl Deiseroff at Stanford, is the first one who tackled this question. And she picked fat to study because very interesting metabolic organ. If you look at a textbook version of how the brain listens and controls fat, you'll get an answer like this, that adipose tissue secretes a hormone called leptin that goes to the brain analyzes this information, and through sympathetic motor function, regulates adipose function. So this is how much fat you should store, when should you burn it, etc. But these in blue, there's also sensory neurons that innervate the fat, and nobody had ever looked at what they do. So you came up with some very unique new methodology to specifically ask, what are the sensory neurons doing innervating fat? And more specifically, what is PIEZO2 doing in this function? And she found that they're absolutely important for regulating adipose function. And I'm just showing you one data point of what happens to fat when you denervate them from the sensory neurons. This is mainly white adipose tissue, and when you ablate it, there's a strong enrichment of beige adipose tissues. So beige and brown fat are more energy-producing fat, not involved as much in storage. So very important biology that wasn't known, and we're very excited to continue this. Interestingly, this phenotype, when you ablate sensory neurons, is very similar, mimics when you're exposed to cold or exercise, and this makes sense, but both of these require extra energy. But there's many open questions. For a soft tissue like fat, what are mechanosensors sensing? We have no idea yet. And also, this was just kind of proof of principle for one tissue type, fat. There's many other sensory organs that we want to study to see what the role of these neurons and mechanosensation is. So everything I told you till now is about neurons, the nervous system. But cells beyond the nervous system experience, sense, and respond to pressure. As you can imagine, your blood vessels, when blood flows through them, experience shear stress. Muscles are stretched. Red blood cells get indented as they squeeze through blood vessels. And almost, again, this was very much ignored in our biology, where most people study chemical senses. The role of mechanosensation was not known. And the sister molecule, PIEZO1, is more expressed in non-neuronal cells. And our lab and many others have been studying what these ion channels do in various systems. I'm not going to have time to talk about it, but PIEZO1 is very important for formation of blood vessels. They're important for regulating bone density. You might have heard that astronauts who go into outer space come back with reduced bone density. This is probably why. Professional tennis players have higher bone density in their dominant arm compared to the other because, again, they feel more pressure on that arm. And it's shown that PIEZO is probably at least partly responsible for this. Also, many roles in both red blood cells and white blood cells that I will not have time to talk about. But I want to tell you about how we're thinking about this. So I think the Nobel came too early because stage one that we're done is mainly looking at all these roles that we knew mechanosensation, pressure sensing was important. And we said, yes, PIEZOs do this. But I'm more excited about stage two, which is investigating roles of mechanosensation in areas that we didn't even think about. So I'm hoping that in 10 years, I'll be able to tell you that pressure sensing is much more important for our physiology and disease than we ever imagined. How do we find new projects? Since PIEZOs are professional mechanosensors, anywhere they're expressed, there's a role for mechanosensation, I can guarantee it. Rose Hill, for example, in the lab found these kidney cells that express PIEZO2. From that, she made this beautiful story of how in kidney they're sensing pressure and are an important part of blood pressure regulation. Clinical data, I mentioned to you already that for bladder filling, this data came from humans, and then we brought it back to mouse to understand it mechanistically. We also use intuition, which might be my favorite of these three approaches. For example, we reasoned that during pregnancy, the uterus expands 50 times, and there's no way that mechanosensation is not going to be important for this. And then again, Ying Zhao in the lab is actually finding very interesting data on this. I want to also talk about pain. So pain is, of course, very related to everything else I've been talking about. Acute pain is essential for survival, but chronic pain is a major unmet medical need. I'll give you some numbers. 25 million Americans suffer from chronic pain. And just in 2019, 10 million people misused opioid drugs and 70,000 people died from overdose. So it's a major issue. So pain is not considered a disease by itself, but it's associated with almost every disease you can imagine. And that's why, like many other diseases, pain, just reporting of pain goes up by age. And stealing a joke from Pete Schultz, our president, I'm at this age right here. It looks like it's kind of plateaus, but I know what's coming. So I want to work on pain to make sure that we try to make a dent in this. Pain is very complex. It starts with detecting noxious stimuli. It could be mechanical, thermal, or chemical. The nerves go to spinal cord. We talked about this, goes to the brain. But in the brain, pain is not just sensory, emotional, cognitive experience. And at Scripps, we want to start a new pain initiative, trying to do more translational research, trying to translate some of this work into future medicines. And we have incredible scientists here, like Hugh Rosen and Ben Cravat, who have drug discovery and pain experience. Together with them, we're starting this new initiative that I'm very excited about. We're also recruiting new pain researchers who specialize in some of the CNS pain to help us discover the future medicines. But the next couple of slides, I want to come back to PIEZO2 and this detection side of pain. So we knew both from mouse models as well as animals that acute pain is not affected in the absence of PIEZO2. So hammer hitting your finger, ouch, is there whether you have PIEZO2 or not. But we're very interested in this pain phenomenon called tactile allodynia. Sounds complicated. Tactile means touch. Allodynia means when touch becomes painful. So you and I experience this when we have a sunburn or an injury. Just touching or wearing a shirt can be painful. But this is actually an aspect of pain that folks suffering from neuropathic pain experience every day, and there's no good treatments for it. And so we asked if PIEZO2 is involved in this. I'm not going to show you, but Shweta Murthy, while she was in the lab, showed that PIEZO2 in mice is absolutely required for tactile allodynia. And then my colleague Alex Chester's lab actually was able to demonstrate this in humans. So this experiment, you take the arm of control or PIEZO2 deficient individuals, and they're blindfolded. So you have this imaginary box A and B. And box B gets things that would cause inflammation. In this case, chili peppers and hot temperature. Using laser Doppler imaging, you can tell that this area, both in the controls and the PIEZO2 loss of function individuals, became inflamed, and it's red. And then you ask, if I touch box B, is it painful? Of course, all the control individuals, 100% can tell that box B and not A is painful. The loss of function individuals don't feel anything. So you tell them you have to guess one or the other. They guess something, and they get it right 50% of the time. The reason this is important is because many pain drug targets have been developed and have been shown to work great in mice, but when they go to the clinic, it doesn't seem to reproduce. So having some kind of proof of concept, proof of principle in human clinical data, as small as this one is great. So now we know that if you block PIEZO2, it might be very useful for some aspects of of pain. And we're using a variety of methods to find ways to block PIEZO2. We're doing this in collaboration with Calibre, screening large libraries. But I also wanted to tell you today about a new project that we're doing in collaboration with Stefano Forli's lab. And this is called Virtual Screen. It's great because the computer does it. And the idea here is that we know the structure of PIEZO2. So we identify pockets where we know small molecules, drug-like molecules, can bind. And we just simply screen using a computer if, in this case, a small 100,000 compound library, if they would bind to these pockets. And in this case, we found 300 hits. And when we actually tested them in the cells expressing PIEZO2, we found a bunch of activators and blockers of PIEZO2. So this is one of the first ones that we identified. We need pretty high concentration to block it in red compared to the black when you stimulate by mechanical force. But this is very exciting for us that you can start from in silico screen and actually find compounds that block it. And again, this was just a proof of concept, 100,000 compounds. We can screen potentially billions. So we know that PIEZO2 antagonists will not be an oral drug where you can take to alleviate this pain, because as I showed you, PIEZO2 is important for so many aspects of touch and proprioception. You don't want to get rid of those sensations. But I think local, topical applications might be possible. And PIEZO2 is not just the only target. Many other peripheral targets could be very promising for pain. I just have one more science to tell you. Piezos are not just in mice and humans. They're actually present in almost all species we've looked at. They're present in worms and flies, and they're present in unicellular organisms and plants. And Ali Reza Musavi, who's also here, who's an alumni of the lab, asked if piezo is important for plants. So plants also sense pressure in many different ways. For example, when their roots experience a hard surface, they have to decide whether to go right through it or go around it. Ali Reza showed that piezo in Arabidopsis, a plant, is expressed very highly at the root tips, exactly where you would want this protein to be to sense this hard surface. And when he knocks out piezo in these plants, their roots start having trouble penetrating harder surfaces, as shown here. In wild type, they go right through this agar, so you don't see them very well. But in this mutant, they go around it, which you can see very clearly the roots of these piezo plants. I say this simply because I think it's remarkable how evolution, once it finds a protein that does something well, sense pressure in this case, uses it for us to sense touch and for plants to sense their soil's thickness or how hard it is. Okay, so this was the end of the science part. I just want to thank current members of the lab, the ones that I showed their data. I mentioned it during the talk. Special thanks to Angela, my assistant, who's been with me for a long time, and I don't know how I would do my job without her. Many alumni, from some of them came back here, and key collaborators, and very importantly, funding by the National Institutes of Health, Howard Hughes Medical Institute, and sources from Scripps, including Doris Neuroscience Center. Okay, Jamie, if you don't mind, in the next few minutes, I want to show some fun slides. And I wanted to reflect on how Nobel Prize has changed my life. And the easiest way for me to do this, since I'm addicted to Twitter, is to dig up old tweets and see how this works out. As you notice, I refuse to call it X for personal reasons. So this might be my favorite photo. This is on October 4, 2021, when Stockholm tried to call me for the prize, but I had do not disturb on my phone, so I didn't get the message. And they're very resourceful, the Nobel Committee, and they Googled Patapoutian, California, and found another number and called my 94-year-old father, who was awake at 2:30. He lives in Los Angeles. He yelled at them for calling them so early. And then, but kind of understood what was going on, so he was able to get in touch with me. And so we pretty much found out just minutes before the official announcement. And Nancy, my wife, who's here, realized and took a picture with my son who came up. And you can see he's touching me. And he doesn't do that often. This is effective touch, and I'm very proud of it. Thank you, Nancy. Okay, Jamie kind of gave this away. You do get a parking spot. That's another huge perk of Nobel. You get gifts. People give you gifts. And so the official Tabasco companies send us about 12 different spicy molecules, kind of activating things because of the research, which we still have some, I think, actually. They last a long time. Thanks to Scripps, they arranged for me to throw the first pitch at a Padres game. Actually, they're playing now. If anyone knows the score, let me know. That was a very special moment, shared with family and friends. But probably the highlight is, I should say that I'm Armenian origin, and the first Armenian to win the Nobel Prize. Also grew up in Lebanon, and again, the first person who's been a Lebanese citizen who won the prize. So I'm very proud of this. I'm going to visit Lebanon at some point, not these days, but I did get to visit Armenia, and they treated me so nicely. And the highlight was they issued a stamp with my. I'm still trying to deal with this. But not everything changes. My grants keep getting rejected from the NIH. And I didn't know if I should tweet this, but I did, and I said, I received another disappointing, unfundable score from NIH. I know I'm privileged. I know nobody wants to hear me complain, but I know a lot of people go through this, and it's very hard for them. So, it still stings. So a lot of people see me fail and talk about it, which is great. Like to share that. And the point of this, though, was that it was a great grant, and it was rejected. And, what's the saying? Who needs enemies when you have friends like Bob? So regardless of this, the stamp and all of that, people around you get worried that you're going to get a very big head. And so my wife, Nancy, has put herself as the head of a committee for ego control. What is it called? Ego management committee. And she does... And she does everything she can together with some colleagues to make sure that I know my place. Okay, and then this is the last slide, and I wanted to share with you, one of the things that happens with a lot of people, you get interviewed and stuff, so you become very introspective and think about things that matter to you and the science. And so I know you didn't ask for advice, and I'm sorry to give you without you asking, but I've come up with some rules that I've learned in science, which I think might be applicable to others too. So the first one is going to be very controversial, because it's don't be too busy. And my logic is that if you're too busy, you're not very creative. In science, that's kind of important. The reason it's controversial is because, a lot of professors here want their students to work very hard, and telling them don't be too busy might not be the best signal you want to send. And I want to say that it's nuanced, right? I worked very hard as a graduate student and postdoc. When you need to work hard, you need to work hard. But there's also time to reflect and think. And my best ideas have come from when I'm running at Torrey Pines State Park, etc. So it's important not to be too busy. What questions should we answer in science? And I think finding the biggest unanswered question, but with a caveat of something that you can reasonably solve in the next 5-10 minutes, 10 years. See, I'm getting ahead of myself. And this is very important, the second part, because if not, all neuroscientists want to solve consciousness, but can you really solve that in the next 5-10 years? So it's a little bit of dreaming of big questions, but with a practical aspect to it is what I found is very good compromise. It's important to prioritize. I think it's as important to quit projects as it is to start them. So, this is my main job with my trainees, is prioritize what they're doing and see which one works, which one doesn't. I've changed fields multiple times, from graduate school to postdoc. Even when I started my lab, I changed fields. And I think that is so useful to do something different, something new, because you bring your whole history and your whole knowledge from another field to a new one. And that's provides good perspective. After giving you all this advice, I have to tell you not to listen to advice, especially if it doesn't make sense, because I got lots of bad advice when I was starting my lab, and thank goodness I had the courage not to listen to them if I didn't think it made sense. Hire people who are smart, efficient, and kind, and please don't forget about the kind. In return, be kind to your trainees, very important. Champion the underprivileged. I feel like lots of people champion me, and when I came from Lebanon, a very naive 18-year-old kid, and there's a lot of people that I owe a lot of gratitude to. Collaborate with people with very different training and experience. Again, this is related to one of the previous points. And at Scripps, I've collaborated with chemists and structural biologists that I know nothing about, and this has really enriched my research program. Surround yourself with friends and family who aren't afraid to call you out when you're wrong. Again, this is very important. I have friends who do this, and my family does this. Although, Nancy, a little bit less would be better. And finally, this is for scientists. Don't forget why you got into this business. Science is super fun. We have fun all the time. And I know we all worry about grants and papers, but try to minimize the noise that causes all this anxiety. And again, I say this with a little bit of being careful, because it's hard to do science these days. Funding is very tough, and a lot of people are on their last grant wondering if they're going to be able to renew it and do the projects. So it's easy for me to come out here and say, have fun. But in reality, even controlling this a little bit, I think helps because that's when good science is done, when you're having fun doing it. Anyway, thank you for your attention.
Well, thanks, Ardem. That was awesome. So what really struck me, let me just ask the first question. What really struck me was that you have these two PIEZOs, but they're in all these different cell types, and they have different jobs. Maybe even the scale of what they have to sense is different in the bladder versus your fingertip. And so you might imagine that that's coordinated in two different ways. So one is just gross anatomy, where things are signaling different parts of the brain, but also you could have other proteins interacting in the cell with the channel that help tune it. And so how does this thing do all of its jobs in different places with different purposes?
A very interesting question, Jamie, because if you compare it to other senses where you were doing in your introduction, olfaction, smell, there's 1,000 receptors doing this. Like, they've divided the job into 1,000 proteins doing this, and we have 2 that seem to be doing most of this. And all of your intuitive things are correct. So Anna Koster, for example, who's here, is just about to publish a paper showing potentially an interacting protein that changes PIEZO's profile. Others have found this too. I think anatomically, how PIEZO in different cells is linked to not just different proteins, but what's the plasma membrane constitution and how they're connected to different cells is going to affect how they respond to this. Also, just between piezo 1 and 2, we don't know exactly what's different about them, but we are starting to find out that they do this slightly differently. That's why perhaps one is used in neurons and mostly in other cell types. So that was actually a big surprise that just 2 proteins can do this vast number of different types of sensing. It seems like they do. Exactly how they do it, we still don't know, but we're investigating it.
So the other thing I found myself wondering about was the patient population. So what's the size of the people who have mutations in piezo, and are they loss of function mutations? Are they protein gets made but it doesn't make it to the surface, or is it just deletion? And are there all of those types?
We have quite a few different types of mutations. So the ones I talked about were complete loss of function. And so the parents each have one missing copy. There's no phenotype in the parents because they have one functional copy, but the progeny, and there's only about, I think, less than 50 people in the world that we know, but it's growing every year. It's a very small population. But on the other hand, we have actually other mutations that we've found that are very subtle mutations in what we call gain of function, so they have more signal than wild type. And those are in much larger populations.
So a question from the virtual audience. And it basically bears on the relationship of sensation of touch and the difference between pain and itch. So what's going on with itch, and how does it relate to pain and piezo function?
So most of, I don't want to get too technical, but most of somatosensory coding is what we call labeled line. I kind of hinted at this in the beginning. So there's a neuron that only senses cooling. There's a neuron that only senses warming. Just like that, there are neurons that only sense itch. And there are other neurons that are specialized to sense pain. So there's some exceptions, but overall, there's a labeled line hypothesis and theory of the sensory neuron that seems to explain most of the phenomena. This doesn't say those don't interact. For example, if you get hurt, you feel pain, you rub your arm. That's not just random. Actually, touch inhibits pain. And so there are lots of interrelationships between the different neurons and how they talk to each other, but they are, at some level, independently regulated.
I'm sure we have a question in the physical audience. Right here, up in the back.
Me? Oh.
Go ahead.
Oh, okay, yeah. I'm kind of curious about the tools aspect, and for people that aren't aware, that we've had a revolution in neuroscience and tools for optogenetics and being able to activate neurons with light, and so it's kind of interesting to think about what you might be able to do with mechanoreceptors with piezo, and so how are you thinking about that, and have you thought about crazy things like extrasensory types of things that are not typically in organisms?
Yeah, so he's asking about the novel neuroscience techniques and how this applies to our research. You mentioned optogenetics, which is use light to specifically activate different groups of neurons. Obviously, very, very useful technology for studying science and biology. As I said, we love to ask questions if they're necessary and sufficient. So optogenetics can say, what happens if you activate this group of neurons? And you can also just silence them and say, what happens if you silence them? But people are also excited to potentially use this in the clinic. It's a bit more difficult. But interestingly, the problem with optogenetics, for example, is that light has to reach the neuron that you will activate. Many of your neurons in the brain, it's going to be very difficult to get light to. And so some people, for example, are working on these mechanosensors and turning into sonogenetics, where you can use ultrasound to activate them, in which case you can then go to a group of neurons that you somehow mark with these sonogenetic markers and then without invasive technology, can potentially activate these neurons. So there's lots of interesting possibilities. Beyond that, just haptics, whether you're playing video game or any other kinds of haptics, including in health. We're collaborating with material scientists as well to think about this, because the only tactile information that we can now do is vibration, and touch is much more rich than that. So learning from biology and taking it to the engineering field is an exciting future endeavor as well.
I have another virtual question. And it relates to the relationship between touch and piezo function and basically mental health disorders. So is there any crosstalk between things like autism and the sensing of touch and pain that is traceable to piezo?
Yeah. Not our work, but there is actually very exciting literature on this now. As you might know, for autism, many kids are very sensitive to touch. They don't like it. And in animal models, David Ginty at Harvard, for example, has shown that this is very relevant to what we're speaking here. And if you have that model only expressed in sensory neurons, you get this sensitivity to touch as well as anxiety and other things by simply expressing the autism gene in these neurons. So what this means and how this translates in the future is work in progress, but very, very interesting indeed.
Great. Another question up in the back?
Ardem, wonderful talk, first of all. It was great. And my question is, it's one thing to modify some of these ion channels with pharmacologic or drug-like compounds, but on the other hand, there seems to be a believable literature about the use of things like virtual reality or cognitive behavioral therapy, meditation that can impact neuropathic pain. And I wondered if you've given any thought to sort of mechanistically how that could be happening.
I'm actually not very familiar with that field.
Okay, I'll have to educate you after the talk.
You said you wanted a new project. Question over here.
You pointed out the types of neurons that can either detect like something like pain as opposed to itching. I was wondering if you know whether the amount of piezo that is expressed on a singular cell level would affect the intensity of the stimuli that a single neuron can detect.
Yeah, interesting. We haven't been able to show that. Traditionally, we've had a very hard time having, I'm going to get technical now, very good antibodies to piezo that we can, so we have RNA information, but we don't have very good quantitative protein information to answer that question. But we certainly see some neurons and some cells have much higher levels of piezo than others, but a functional correlation with that hasn't been done yet.
Okay, I think we're a little over. We could go on, but it's all good. So thank you for your words. Thank you for the time. Good science, good words to think about for life. We do have another front row, it's November 6th, and it's Danielle Grochon. She's one of our young assistant professors. I actually collaborate with her, but she's going to be speaking about mitochondria and mitochondrial stress. Invite you to join us out on the patio for a reception. But if anything else, I invite you to come back. It's a great experience to come in for the real thing. And when you do, you'll find me in the front row.